environment and stress. So I will be chairing this session. I'm Mahasweta Saha from the University of Essex. And so I would just request all the speakers to please stick to the time. And I will be using certain alert symbols. So with this, I would like to welcome our first speaker, Ben Sheldon, who is going to talk about spatial scale governs selection on breeding rate in a wild bird population.
Okay, thank you. Okay, great. Can you hear me at the back? Yes, I think you can. So what I want to talk to you today is about some work we've been doing and ways of trying to think about the determinants of selection on breeding date in wild bird populations. And this is a sort of a classic old problem. So typically what's observed is that early breeding attempts produce more surviving offspring. And this is a classic sort of case of this paradox of a trait for which there's genetic variation, which we know to be the case for breeding time, and selection on that trait, and that trait apparently not at its optimum. And there are many other examples like that from evolutionary ecology and wild populations. Often the patterns with breeding data are particularly sort of strong and robust. So this figure here just shows, don't worry about the y-axis, sorry, don't worry about the x-axis here. The y-axis just shows the distribution of selection differentials in 44 years from the white and great tit population. 42 out of 44 years, the selection differential is negative for this trait. And those cases where it's positive are very weakly positive. And there have been lots of explanations for this paradox, and perhaps two of the main explanations, one proposed really by Chris Perrins back in 1970, is an argument based on optimality. The idea there is that food is scarce early in the breeding season, and there are costs to do with breeding early, and if food is scarce, those costs are too high. So optimality argument suggests you should breed later, or the population mean should be later than for some individuals. The other is a sort of argument based on quantitative genetic principles, outlined particularly by Price et al. In 1988. The idea here is that actually that the selection on breeding time might be illusory. It simply might result from a covariance between, say, maternal condition, breeding time, and other fitness components. The point of this talk is to sort of try and suggest that there's a sort of slightly neglected dimension here, which is thinking about the spatial variation within populations, and this creates a slightly different picture when it comes to understanding selection on breeding time. And the context of this study, I'm going to talk about data collected as part of the long-term study of great tits in White and Woods, started in 1947, and using a very standard protocol for collecting data on the timing of life history events and the fitness consequences of those events. And the reason that we think breeding time is interesting in this system and why it's under selection has to do with birds timing their breeding to coincide with a peak of food availability in the environment. So this is a classic story here that we have deciduous trees like an oak tree. The leaves come out on those trees and they're fed on by a guild of Lepidoptera, particularly the winter moth. Those caterpillars hatch and feed on the newly emerged leaves, and birds have to time their breeding to coincide with the peak abundance of these caterpillars in the environment. So we've got caterpillars hatching to match the peak in the bud burst of the trees. Those caterpillars grow over some period of time, and the birds have to time their breeding so that the peak of food demand for their offspring coincides with there being lots of caterpillars around in the environment. And the problem for them is that they have to lay their eggs about 30 days before that peak of food demand of the offspring, so they've got to, in some sense, anticipate or time their breeding correctly to anticipate that peak in food demand. And the problem, of course, and this has been studied for some time, is that the timing of these events varies from year to year. And we know quite a bit about the causes of that timing at the level of the population. Large-scale climatic factors predict the relative timing of these peaks. And both birds and caterpillars seem to respond in the same way to these climatic factors. And the result of that is that over time, the timing of breeding of birds and the half-full date, which is the peak date of caterpillars in the environment, are closely synchronized. So there's a pretty good correlation with those at the population level from year to year. So each of these dots here represents the mean for a population, could be several hundred birds there. And of course that's a bit problematic when you think about what a population really comprises. So what we're doing here is effectively ignoring all of the variation that might occur within this is White and Woods here, 385 hectares. Each of the dots here is a nest box that could have a great tit breeding in it. We know from studies of foraging behaviour of these birds that they only exploit very limited areas around the site where they're breeding. Of course, they don't fly all over the woods to collect food. That wouldn't be energetically very sensible. So really, they use a limited amount of space in the environment, and we should expect them to be sensitive to fine-scale variation in the environment. And what hasn't really been the case when people have been thinking about selection on breeding time is how spatial variation at the level within populations affects selection on phenology. And if we look at all of the data collected from this study over time, so each of these individual nest box sites has been fixed over time, we can characterize each site as whether it's relatively early or late relative to the rest of the population. So if you do that, you get a pattern like this. So the size of each dot is proportional to how frequently it's been used by great tits over the, there's about 54 years of data analysed here. And the coloration is proportional to whether it was relatively early or late compared to the population means. It's standardised by the mean in each different year. And there's lots of structure present in this system. So if we look at the sort of the Mantel correllogram, you can see that sites that are close together tend to be more similar to each other. Sites that are further apart tend to be less similar. But actually there's variation at multiple scales here, because you can see there are areas where pretty much all the birds are early, and where all the birds are late. And this tends to be related to large-scale effects like altitude. But then there are other areas where there's fine-scale variation. So even over relatively short distances, you go from areas where birds are early to where they're late, over just a few hundred meters. So we've been interested for a while in trying to understand how this fine-scale variation comes about and what its consequences are. And to do this, we carried out a study of the phenology of individual oak trees within a particular part of Wytham Woods, where we scored the development of individual oak trees as budburst occurred for 117 oak trees rather, and for 44 of these we also measured the half-full date of caterpillars under those individual trees. The nice thing about oak trees is they don't move around, so they're in the same place every year reliably. But also, if you go in repeated years, what you find out is that individual trees are extremely consistent in their budburst date. So the mean might change from year to year at the population level, but the relative rank order of budburst date for individual trees is very, very consistent. And if you plot the variation in budburst date for individual oak trees, you get this sort of map here. This is a heat map where red is relatively late budding oaks and blue is relatively early budding oaks. And successive years show very similar spatial patterns. So the correlation between these patterns is really very high. And these patterns also predict the timing of caterpillars on those individual oak trees. So individual oak trees vary in their budburst state, and the caterpillars, the timing of caterpillars on individual trees is predicted by the budburst state of individual oak trees. So what we wanted to ask first of all was whether there's any evidence that great tits synchronize their breeding in relation to these local environmental effects as well. So we used long-term data from 1965 to 2009 for this site where we collected this data. And we asked, basically we did a bunch of sort of model fitting where we asked what spatial measures of phenology in oak trees were the best predictors of tit timing of breeding. And I won't go into the details here, but basically there's lots of different ways you can characterize that spatial variation based on heat maps or different buffers or just distance to nearest trees. Basically, they all consistently suggest that oak trees within 50 meters predict the timing of laying for individual great tits. So great tits are very sensitive to this small-scale variation in the environment. So why do they show this spatial matching? So one possibility is, of course, that it represents a sort of an optimal timing in response to local conditions. And we can test whether that's the case by using the fact that we've got repeated observations at the level of individual nest sites. And we can separate those, we can take those observations and separate the variation into two components. One is just the next nest box average date, where any associations between date and fitness represent the effects of the average properties of that location, or covariance between female quality and site quality. But more interestingly, the deviation from that mean represents the direct effect of variation in the match to the local long-term mean at that site. So just to illustrate this, this is one individual nest box, extra 35, which had extra 55, which had 35 breeding attempts by 28 females over that period of time. This is the number of recruited offspring in relation to the deviation from the nest box mean for that site. And so this is just showing the quadratic relationship that describes these data with a peak that's very close to 0 for this box. And we can do this for all of the sites where we've got multiple observations and look at the different fitness components for the birds breeding in those different sites. So for example, if we take here the measure is the probability that a breeding attempt is successful, in other words, whether it fledges any young at all, in relation to the deviation from the local, the nest box specific mean laying date for that individual site. And we've got about 500 nest boxes where there's 15 or more observations for each boxes. And correct for various random effects, of course. And what you can see for this variable is that there's a strongly quadratic relationship between, there should be data, for some reason the data plotted on that haven't shown up. I can't see them at least, so that's weird. There were data plotted on that when I created the figure, but there's, this just shows the model fit. That's what all you can see. So you can see there's a strongly quadratic relationship between this deviation and the relative measure of success. And the same is true if we look at other measures of breeding success, where these measures are defined as occurring before fledging happens. In other words, these are measures of fitness that are determined by processes that should be local in their operation. So for example, if we look at, we've already looked at the probability of success. Here's the number of offspring fledged. Here's the proportion of eggs laid that were fledged offspring. This is the fledging weight of the offspring. In each case, there's a strongly quadratic relationship with a maximum that's quite close to a mean of 0, in other words, to the average laying date for birds in that individual nest box. If we look at a measure of fitness taken after dispersal happened, we see a different kind of pattern. So this is the pattern in terms of the numbers of recruited offspring in relation to the deviation from the local, the long-term mean for this nest box. You can see that the peak is shifted quite well towards negative values. In other words, breeding attempts that are earlier than average tend to have the highest success in terms of numbers of recruited offspring. So what seems to be happening here is that we've got two kinds of patterns. So when we have fitness components that are likely to be determined by local processes, so in other words, processes that occur before the offspring leave the nest, it appears that we've got evidence for sort of local optimization of breeding time. In other words, if you breed at the time that is the average time observed for that individual nest site, that's when you have the highest relative fitness. When we're talking about fitness components that are likely to be determined by processes occurring at the population scale, so after dispersal away from the breeding site, it looks like we've got directional selection for breeding time. So how can we combine these two different perspectives? Well, on average, if we're looking at in terms of local conditions, birds seem to hit the correct optimum for those local conditions. If we have a population where there's spatial variation in that optimum, in the timing of that optimum, what that's going to lead to is within population differences in birth date after dispersal. So after birds have dispersed and start interacting with individuals from different parts of the population. And it's quite plausible, and there's some evidence from other studies, that having an early birth leads to a competitive advantage in settling down to breed as an adult yourself. But the problem is, from the point of view of an individual great tit, birds are facing a multi-scale optimization problem. So timing locally is important, but also your timing relative to other individuals in the wider population is important. But there's inequality in the amount of information available. You can have very good knowledge about what the optimum timing might be for your local site, but often you will have very little knowledge about your timing relative to the entire population that your offspring might be competing against. And indeed, there's no really straightforward expectation for how the variation in that optimum at the larger scale should be distributed. And I think it's interesting to think about this tension between small scale and large scale optimization and how that feeds through to determine the optimal pattern for timing of breeding in birds. Okay, so thanks very much.
Thank you. So we have time for one quick question. Please.
In the forest layout, the very first figure that you showed, one could clearly see that
Possibly, although I think the scale is probably larger than those edge effects would tend to operate. I think we're talking about over several hundred meters for those. I mean, I think I would only expect those effects over relatively small scales relative to the size of this population. So I think I don't think edge effects will be important for most of the birds in that population. But one of the, clearly in a sense, one of the points here is that the geometry and physical characteristics of a population will determine things like how much variation there is in the optimal breeding time at the small scale. Of course, when those birds are combining to compete for sites to settle, that can generate variation in, for example, birth date. So, yeah. Okay.
Thank you very much. Now, talking about dynamic changes in habitat quality, modulate heterogeneous responses to climate change in a wide bird population.
Not the most catchy title.
Yeah.
Yeah, so today I'd like to talk a bit about some work looking at factors that might explain variation in response to climate change in wild great tits. So in seasonal environments that are characterised by a transient peak in resource availability, animals that can time their reproduction to coincide with this peak in resources will maximise their reproductive success. And as Ben showed in the previous talk, timing of breeding can actually be subject to quite strong natural selection. And in line with this, over recent decades, as climates have become warmer and springs have been shifting earlier, we've actually seen lots of examples of species shifting their life history events earlier in time. But not all species are actually responding in the same way. So there's evidence from both freshwater and terrestrial systems that the different trophic levels are actually adjusting to climate change at different rates to one another. So in terrestrial systems, you can see that it's actually the lower trophic levels that are showing the most rapid rate of change in response to climate. And this is leading to concerns that trophic mismatch might actually become quite widespread in these systems. So as Ben outlined in the previous talk, the tritrophic system of passerine songbirds feeding on caterpillars that in turn feed on the emergent leaves of oak trees is actually an excellent system for looking at questions relating to adjustment to climate change. And work from our long-term study population at Wyton Woods near Oxford has shown that British tits are actually tracking climate change very well by plastically adjusting their timing of breeding between years. So in our population, both the caterpillars and the tits are very sensitive to spring temperature. And this means that as springs have been getting warmer over the last sort of 55 years, the tits and the caterpillars have remained synchronized in their spring timing. But this is not actually the case for all tit populations. So trends from another long-term population study of great tits in the Netherlands are actually showing that although they're kind of experiencing the same sort of climate change as the Wyton great tits, although the caterpillars are shifting their spring timing earlier over time, we're not seeing this in the great tits. And this is leading to trophic mismatch developing between these species. So what's causing this sort of different response in the Dutch great tits? Well, a likely explanation is the fact that climate change is not actually consistent across the whole of spring in the Netherlands. So temperatures during early spring, which is when great tits are actually making their decision of when to start breeding, actually hasn't changed over time. But later spring temperatures, which is what actually determines how quickly the caterpillars grow and therefore the timing of this peak in food abundance for the great tits, has increased significantly. And this means that there's actually a mismatch that's developed between the temperature cues used by great tits to time their breeding and the temperature during the period where selection is actually acting on this timing of breeding trait. So comparing population responses can provide us with important sort of information about how the sorts of things that might be constraining response to climate change. But can we also gain insight from looking at variation over smaller spatial scales? So in the previous talk, Ben kind of showed that just looking at population means actually can mask interesting and important variation that's occurring over a smaller spatial scale. So this figure just shows the population response of great tits to climate change in White and Wood. So you can see that as springs have become warmer over time, springs have become earlier over time, great tits are now laying about 2 weeks earlier than they were in the early 1960s. But are these means concealing interesting spatial variation? Well, when we actually look at this, we see that there is indeed significant variation across the population in the rate of change that birds are adjusting their timing of breeding. So each dot here is an individual nest box, and the colour shows the rate of change in laying over time. So red boxes here are boxes where laying has changed at a faster rate than the population average, so have been getting earlier. Orange are boxes that are sort of around the population average, and blue are nest boxes where laying has actually changed slower than the population average, as just shown by this little schematic here. So you can actually see that there is sort of spatial structure in these data. And when this is kind of formally analysed, there is significant spatial autocorrelation. So this plot here just shows that at distances, so nest boxes that are within about 750 metres of one another are more similar in their rate of change in laying over time than those that are the further apart. So what could be driving this variation? Well, we know that all of these birds are experiencing similar global temperature cues to each other. So it seems unlikely that this variation is caused by the sort of temperature mismatch that seems to be constraining response to climate change in the Dutch great tits, as I was explaining earlier on. So could it actually be that the caterpillar and the oak phenology is also varying over a similar sort of spatial scale in their rate of change over time? Well, while we can't really rule out this possibility, we do know, as Ben was explaining in his talk, that oaks, individual oaks are incredibly consistent over years in the timing that they come into bud. So these figures just show heat maps of oak phenology in one area of Whiteham in 2 consecutive years. So you can see that these patterns, these spatial patterns of oak phenology are incredibly similar. We also know that in Whiteham, at least, the phenology of oak trees is not related to tree age, and there's very little turnover of individual oaks at the sort of timescale we're looking at. So this suggests that the optimum time to breed is actually likely to have remained quite stable for individual sites over time. So another possibility is this could be driven by change in habitat over time that could actually be constraining birds' ability to track changing climates. So we know that oaks are a key component of tit habitat, so they harbour a lot more caterpillars than any other tree species. And work in our population shows that tits breeding in very oak-rich areas will bring more caterpillars back to their young and will also fledge chicks in better condition than those nesting in oak-poor habitats. So we were actually able to look at territory sort of quality change over time using data we have for Whiteham on the oak trees. So in 2011, we mapped all of the oak trees in Whiteham, so it's just shown by the green dots here, so nearly 6,000 individual trees. And when we did this, we also assessed the health of each individual tree by just estimating the amount of canopy that was alive and healthy. So oaks can suffer from something called chronic oak dieback, and this is thought to be caused by a sort of complex interaction of damaging abiotic and biotic factors, so things like recurring droughts, and strong winds, and also opportunist attacks by sort of disease and insects and things. And this can cause crown thinning, but also complete tree death. And this is what the health data looks like for Whiteham. So each dot here is a tree, and the colour just indicates the health. So green trees are healthy, and dark blue are unhealthy trees where more than half of their crown has actually sort of died. So we wanted to just confirm that this was actually a measure of decline over time rather than just fluctuations in health. And to do this, we just looked at the survival probabilities of 395 oak trees across the woodland that we knew were alive in 1975. And we found that the probability that they were still alive in 2014 was strongly predicted by the local oak health. So trees that were surrounded by unhealthy oaks were more likely to have died. So we can be pretty confident that this is indicative of sort of decline in health over time. So going back to that plot, you can see that there are areas of the wood where it seems that the oak health has declined over time, and therefore the tit habitat quality will have declined as well, and other areas that have remained relatively stable. So our question was really, does this have consequences for the great tits? And the answer seems to be yes. So we see a relatively strong correlation between the rate of change in laying over time and the health of the trees surrounding a nest box. So the y-axis here just shows the rate of change, where more negative numbers show a faster than average rate of change. So you can see these nest boxes with fast rates of change tend to be in sort of healthy areas compared to boxes with slower rate of change, which are in areas with more oak dieback, so areas where the habitat quality has actually been declining over time. So this really does seem to suggest that tits nesting in areas where habitat quality has declined may actually be less able to keep up with advancing springs. And this could be because they actually just can't gather the resources they need to begin laying at the appropriate time, or something about the change in the habitat is affecting the cues they're using to time their breeding. So yeah, this is something that needs sort of further exploration. So just to quickly sum up, we see variation in response to climate change between species and between populations, but we also see this between sites within the same population. So this sort of variation can actually occur over a surprisingly small spatial scale. And we show that this variation in great tit response is actually linked to spatial heterogeneity and habitat change over time, which could be due to habitat quality actually limiting these birds' ability to keep track of changing climates. So this work really shows that measuring or looking at phenological change and also resource utilization at a scale that's relevant to individual animals can actually provide quite sort of powerful insight into the constraints governing the extent to which animals can adjust to environmental change. So that just leaves me to thank Ben, my collaborator, and also the Whiteham field workers who collected all of the long-term tit data, and NERC and the Edward Gray Institute for funding. Thank you.
So we have time for some questions. Anybody?
Thanks so much. Is it possible that there's any cultural transmission of egg laying between birds, or does that not make sense?
It is something that, in terms of kind of gaining social information, it's definitely something that could happen. We know that the great tits do actually, they're quite nosy during the breeding season early on, and they'll go and they'll look in each other's nest boxes. So there's certainly potential for them doing that, but we've not really looked into that much yet. But it could explain some of that, sort of the spatial patterns, yeah.
Anybody else? Oh, well, if not, I have a question. So, do you think it's phenotypic plasticity or rather adaptation?
So we know that it is phenotypic plasticity. So we've looked at this by analysing sort of individual responses, so birds that are bred in multiple years in different conditions. And you can see that when there are big temperature differences between years, females are actually adjusting to that. So it seems like the whole response we're seeing is actually...
How quick is this? How quick is this change? So how quickly they respond? I'm entirely out of this field, so my question...
So I mean, they're responding immediately. So literally, a bird that breeds twice in 2 consecutive years can match the conditions sort of perfectly.
Okay, thank you very much.
right, thank you. I'm going to continue on topics such as environmental variation and how that affects the life of animals. So as we know, most organisms live in varying environments. So basically there's short-term variation in weather conditions, but also large-scale patterns such as habitat loss and fragmentation, the spread of invasive alien species, and climate change. So very large changes and problems for many organisms. So the really important question for us ecologists is how can we understand and predict these changes? And one approach I've been using is the resource allocation framework. So trying to understand the mechanisms that happen when individuals are faced with changing conditions and changes in the available resource pool, and how they then allocate these resources to different processes. So oftentimes we think of reproduction and survival as the sort of only possibilities for resource investment, but actually the individual has many more choices, including those that include investing energy into movement to find more resources or to escape hostile conditions. And the sort of really interesting question I'm focusing on is what happens when the available resource pool changes, or what happens when the decisions to use energy change the relative and absolute fluxes of energy. So in this talk, I'm going to focus on two types of stress, one being food stress, so the lack of food periodically. And in the case of butterflies, my study system increased flight. And as we know, flight is a very expensive way of moving, so it's consuming those resources that could be used for other life history traits. In this talk, I'm going to present work done using 3 butterfly species, 2 North American species, Coleus eurythemi and Spirea mormonia, and the European or Eurasian glanville fritillary butterfly Melita kinxia. And in this talk, I'm going to focus on just 2 life history traits, fecundity and metabolic flight capacity. So the methods we used for manipulating both resource intake, energy intake, and increased flight are depicted here. So basically, we had a split brood design, and in our food restriction experiment, we gave female butterflies half of the volume of sugar water consumed by their sister in the control treatment. In the flight stress experiment, we fed them all with as much sugar water as they wanted, but the individuals in the flight treatment were forced to fly 3 times 4 minutes every day. And we then measured a number of life history traits. So if your favorite life history trait isn't mentioned in my talk, chances are that we actually did measure it, so come talk to me later. So how to measure flight capacity, which is a rather vague trait, I would say. My favorite method is the measurement of flight metabolic rate. So basically, a butterfly is flying in a respirometer, and in real time you can see its CO2 output. And these 3 figures here show 3 individuals that show very different flight characteristics. The bottom individual being basically a hopeless flyer. So look at the results. So how was fecundity affected by food restriction? Here on the left, we have data from Coleus eurythemi and Spirea mormonia, and I've plotted the total number of eggs laid by individuals in the food restriction experiment. So we see that in the case of Coleus eurythemi, egg production went down by approximately 25%, whereas in Spirea mormonia, the decrease in the number of eggs laid under food restriction was over 50%. So when we gave them 50% food, they laid only 50% of the eggs of the controls. In the case of increased flight, we see a different pattern. There actually was no difference in the number of eggs laid, but the timing of oviposition changed. So individuals forced to fly were laying eggs earlier than the control individuals. So looking at older work, we could actually predict the pattern seen in the reduction in the number of eggs laid based on knowledge on the reproductive biology of these species. So butterflies differ in the ratio of larval versus adult-derived food that is used for producing eggs. And we actually knew that Spirea mormonia is using up to 80% of adult food-derived carbon to build its eggs. Therefore, the pattern we saw that the reduction in egg production was larger in Spirea is sort of what we predicted. So what happened to metabolic flight capacity, flight metabolic rate? First of all, here on the left we have the results from the dietary restriction experiment. We see that there is a pattern of sort of senescence, lower flight metabolic rate as the individual ages, but there was no effect of the food treatment. I should also point out that these metabolic rates have been adjusted for the size of butterflies, which is one of the main factors also affecting metabolic rate. On the right, we have the result from the flight treatment experiment. And again, we see a similar pattern overall. But what's interesting is that there was this interaction between age and the flight treatment, suggesting that individuals that were forced to fly were losing their ability to fly faster. So they were senescing faster than the control females. So I wanted to know how general these findings really are, and translated these same questions to another study system, the Glanville fritillary butterfly study system, which has been studied by Ilkka Hanski and colleagues in Finland. And obviously, these butterflies are distant relatives, and there are many kind of differences in their life histories and the places where they live. One of these differences being that Melitea lays clusters of eggs, whereas both Coleus and Spirea lay single eggs. So I performed an experiment where I combined the two previous experiments. So we had control butterflies, we had individuals that were forced to fly, and then we had individuals that were forced to fly under food stress. And again, we collected all the eggs and measured the metabolic rates and a number of other life history traits. For my own sanity, I only performed 3 measurements per individual. So a look at the fecundity results. Here on the left, we have the total fecundity in these 3 different treatments. So what we see is, interestingly, stress, in this case, flight stress or flight with the added food stress, caused individuals to lay more eggs than in the control. Note that there were quite a few individuals that did not lay any eggs, and that too, that number is higher in the control treatment. So it seems that in this species as well, flight promoted egg laying. And we can also see on the right-hand side how the age at the first egg-laying event also seemed to follow stress in the way that stressed individuals started laying eggs earlier. With flight metabolic rate, we saw again a pattern where metabolic rate is decreasing with age, But interestingly, here there was no differences among the different treatments. So the pattern of faster senescence among flight-treated individuals didn't seem to be the case with Melitea kingsia. So to sum up these findings, first of all, one of the main findings was that flight seemed to be conserved under all kind of stressful conditions. So even when the butterflies have less energy available, they still don't change their flight metabolic capacity. And I think that's reflecting the fact that butterflies really need flight to survive. So flight is a really important fitness trait. So therefore, they seem to conserve their flight capacity. And also for the obvious reason that being able to fly gives you the chance to escape unfavorable conditions. The finding we saw in well, basically both Spirea and Melitea was that flight had a positive effect on fecundity, is a really interesting one, especially in the case of, in the last experiment I showed you with the glanville fritillary butterfly, where the control individuals were the worst egg layers, which perhaps could suggest that there is some sort of a couch potato effect. So if you don't move, you don't reproduce. This is interesting in the context of earlier work on the glanville fritillary, where it has been shown that individuals from new and old local populations in this classic metapopulation differ in their flight capacity, and individuals from new populations being more mobile and also being able to have higher fecundity and invest in reproduction. This is sort of interesting because much of the life history theory has been focusing on the trade-off between fecundity and flight. And there are some very nice examples from crickets, for example, that have a very strong trade-off. However, I was looking at some older literature, and to my surprise, there actually seems to be a nice literature on the connection between flight and egg production in insects. So I think it's really something to look into, the hormonal control of egg production and all that, and not to just simply think of terms of trade-offs. So we don't know the exact ratios of Melitea kingsia, the glanville fritillary, how much it depends on adult feeding for its egg production. But based on these data and other work, it seems that Melitea kingsia is at the low end of this spectrum. So they rely more on larval resources for their eggs. So overall, butterflies are very sensitive to environmental changes, but these effects are often species-specific. And it's also interesting to note that flight perhaps isn't that bad for butterflies after all. But we know that, for example, increasing habitat fragmentation that requires more and more flight at some stage will drive populations extinct. So too much is too much when it comes to fragmentation. All right, I want to thank my co-author Carol Boggs and of course you for listening. Thank you.
So we have time for one quick question.
Can I just ask how many egg clusters Melitea lays in the wild?
So we know from these cage experiments, we have this sort of large 30 by 30 meter cage where they are sort of under semi-natural conditions that typically, what the average number is, I don't know, but I would say it's about 4-5 egg clutches of maybe 150 up to 200 eggs.
Okay, so given that you measured the first three clusters, couldn't it be that in the case of stress they're essentially just producing more eggs earlier, but they're still be producing a lower number overall?
Yeah, that seems to be the case, actually, yeah. Of course, from the perspective of a butterfly, which tend to be rather short-lived, anything you do in early life is going to be important.
Thank you. So our next speaker is
Eleanor.
Eleanor Gibson-Forty, and she's going to talk about herbivory along steep rainfall gradients. Intraspecific patterns contradict the resource allocation hypothesis.
Thank you. So today I'd like to talk to you about plant-herbivore interactions along a resource gradient. Whilst there are many theories to predict plant-herbivore interactions along a resource gradient, the Resource Allocation Hypothesis, or RAH, first put forward by Coley and co-workers in 1985, has enjoyed much empirical support, mainly through interspecific studies. And this theory posits that changes in resources along an environment select for different plant growth rates. All the while, herbivore pressure is assumed to remain constant. So, in low resource environments, such as arid environments, shown here, you have low resources and therefore a low plant growth rate. And then as resources become higher and the environment becomes more favorable, the growth rate of plants becomes higher also. And this is related to and is correlated with the investment in chemical defenses. And so in low resource environments, you have high chemical investment to be able to protect your expensive resources. And this steadily decreases as you move towards the more productive environments. And we can really see this in the real world. So here on the left, we have a plant taken from an arid area in the desert. You can see it's very small, it's not growing very fast. The scale here is my thumb. And then we move over to the wetter area. This is in a Mediterranean climate. The plant is much larger. We can see here the scale is my foot. So we can understand quite easily that 1 gram of herbivore damage in this plant is a larger percentage of its net production than in the Mediterranean plant. And therefore it is expensive to replace resources lost to herbivores. So in this context, we can say that plants in arid and low resource environments are limited in their growth by their resources. However, as we move over to the more Mediterranean environment, resources are much more abundant, and therefore these plants tend to be limited by competition. And so they're investing in chemical defenses actually reduces their absolute growth. And the RAJ, the resource allocation hypothesis, can be shown graphically. So here we have our resource gradient along the bottom, and we can see our increase on the y-axis of leaf damage, whilst our chemical defenses decrease and herbivore pressure remains constant. So talking about a resource gradient, I've defined it here as the amount of an essential component for supporting plant life changes in space, and I've named but just a few here. And resource gradients are an interesting framework to use because they allow us to study climate change and climate variation, because often they reduce confounding factors. So often one element remains constant along the gradient, such as the bedrock. And therefore, as plants are distributed along these gradients, we're really able to study the fitness and the evolution of plants by looking at local adaptation, both at the interspecific level, so between species, and at the intraspecific level, so within species. The resource gradient which I was working on is a naturally occurring rainfall gradient in the eastern Mediterranean basin. And in these Mediterranean mid-latitudes, water tends to be the main limiting factor for ecosystem functioning. And interestingly and rather unusually, global circulation models agree in their predictions for future decreases in rainfall. And so whilst much work has shown that the resource allocation hypothesis does happen in interspecific studies, little work has been done on the intraspecific patterns. And this has led to the key questions of my experiment, which I'll just explain briefly now, starting at the top and moving clockwise. So the key question is, should the patterns be the same between interspecific and intraspecific? And specifically, do plants and herbivores interact differently along a rainfall gradient? Can the environment change biotic interactions? And the environmental change here is rainfall. So can rainfall decouple this relationship? And lastly, is there a trade-off between growth and defense in different environments? This question is often very difficult to conclude in interspecific studies because the species identity itself is a confounding factor. So a little bit more detail on my methods. I worked on a total of 13 sites in the Eastern Mediterranean Basin, ranging from arid, shown in the open red circles, semi-arid in the open yellow circles, Mediterranean in the open blue circles, and Mesic Mediterranean right at the top in the open purple circles. And you'll notice that the colours of the circles are also different. And this is because I had 3 geographical replicates. I used 3 different gradients in these areas. I used a south-north gradient shown in black, an east-west gradient shown in blue, and a west-east gradient shown in green. I used 14 different species of the annual Brassicaceae family, and these species were distributed along the gradient. I used this species because glucosinolates are known well to be one of the main chemical defenses. For the study, I selected a maximum of 50 individuals per species per site, and from these individuals I then selected randomly 1 fully expanded green leaf to perform the observation on. I then estimated leaf damage as a percentage of total leaf area and also observed the presence or absence of insect enemies on the plant. So when we look at the results taken together at the interspecific level, pooling all the species together, we really do see evidence to support the resource allocation hypothesis. On the x-axis, on both graphs, we have average rainfall ranging from arid at 90 millimeters over here to Mediterranean of 1,000 millimeters on the other side. And here we have leaf damage across the rainfall gradient and enemy presence across the rainfall gradient here. And we can see quite clearly a significant increase in the amount of leaf damage as we go along this resource gradient. All the while, we have our enemy pressure remaining constant, as is assumed in the hypothesis. And so we can partially support the resource allocation hypothesis by seeing that we have this increase in leaf damage whilst herbivore pressure remains constant. However, we can't fully support this hypothesis as yet we do not have full chemical analysis of the glucosinolates, and we will need this to fully confirm. But the question throughout is, are the patterns same at the intraspecific level? And so I'm going to show you results from 7 different species. And in these 3, we do have support for the resource allocation hypothesis. We see this increase in leaf damage, whilst enemy presence here decreases, but this shows that it's the resources that are driving the investment or the damage that's happening. However, when we look at other species, we see a different mechanism at play, and therefore we reject the resource allocation hypothesis. In Araucaria pinnata, we see an increase in leaf damage with an increase in enemy presence, and the opposite trend seen in Alyssum simplex. So from this, we can suggest the mechanism behind is that the investment in chemical defenses remains constant across the gradient. So with more herbivore pressure, you get more leaf damage, and therefore this rejects the resource allocation hypothesis. And then lastly, in 2 other species, again, we see a different relationship. In Biscuitella didyma and Hirschvardiancana, we see a constant leaf damage across the gradient with an increase in enemy presence. Again, a different mechanism must be at play. We reject the resource allocation hypothesis. And so in these last 2 species, we ask what's happening. Is it that there are more chemical defenses in wetter sites to tolerate more herbivore pressure? If so, we suggest that herbivore pressure selects for chemical investment. Or it could be the other way round. The resources are limited in drier areas, and so defenses are also reduced, and therefore we suggest that the environment selects for chemical investment. And previous work on Biscuitella didyma on this gradient looked at the glucosinolate concentration, and this showed an increase from the arid to the wet. And so these results, together with the novel results from my experiment of this constant leaf damage with an increase in enemy pressure, allows us to suggest that actually it is more chemical defenses in wetter sites to tolerate more herbivore pressure. And so we could say that for this species, we can accept the hypothesis that herbivore pressure selects for chemical investment. And so again, it goes against the resource allocation hypothesis. So bringing it all together, what can we say from this experiment? Well, we can say that at the interspecific level, the resource allocation hypothesis is supported. We saw this increase in leaf damage whilst herbivore pressure remained constant. And for 3 out of 7 of the species that I worked on at the intraspecific level, we also had support for the resource allocation hypothesis, increasing leaf damage across the gradient. However, for 4 out of 7 species, for more than 50% of the species, we couldn't accept this hypothesis, suggesting that there are many other mechanisms going on here, and that there are different intraspecific theories to answer this. However, it's impossible for me to do so at the moment because I don't have the concentrations of the glucosinolate. And this is the next stage of my PhD, to be able to take these concentrations to fully explain the relationship between plant phenotype, that's the leaf damage, the environment, the change in rainfall, and the herbivore pressure, the enemy presence. And I'll do this in a cost experiment looking at constitutive glucosinolates. And so the main take-home message of my research to date is that for some species at the intraspecific level, the environment selects for chemical investment, whilst for other species, herbivore pressure is the main selective pressure. And so care must be taken when generalizing from the interspecific level to the intraspecific level. And with that, I'd like to thank the funders for their money, the people for their help, and you for your time. Thank you very much. Absolutely. So the herbivores that I was looking at here was insects and pathogens, because often the damage performed by insect and pathogens are quite hard to differentiate. I wasn't looking at all at grazers or larger herbivores. And it must be noted that there are a lot of grazers in these areas. There's a lot of goats and other sorts of animal herbivory. Oh, I see. All of them were taken together as damage. I didn't specify. Yes. No, this wasn't available at this time. My expertise did not span that far at that time. Yes, absolutely. That's the idea. So I took 50 individuals of plants, and on each plant I observed whether there was a presence or absence of herbivores. So if it was a presence, it was 1. If it was an absence, it was 0. And then for the 50, it was summed. So if there was 25 herbivores, that was 50% of that species per that site.
You identified herbivore because there will be big differences between monophagous and oligophagous. And big differences, monophagous on arid areas and oligophagous with high diversity in wet areas.
So a similar question to the first one. I wasn't able to separate between herbivores during this experiment. So I don't know specialists or generalists or oligophagus or polyphagus or minors or anything. And this is, yeah, it does, it is a small problem with the design.
Your first figures will be resulted by this difference between monophagous and oligophagous.
Yeah, absolutely. And we are thinking of other ways in the future to be able to build on this, to go into a second year into the field and to really tease apart what sort of herbivory is going on. So this is definitely in the pipeline for future work.
Any more questions? Yes, please.
Do you think, sorry, what's the phylogenetic relationship among the different species within the family that you're looking at? And does that match to that general pattern? Or do you think you catch it within species-specific stuff?
Well, this is a good question. Thank you very much. I don't actually know the specific relationship genetically or phylogenetically between the species. All I know is that they are within this family of Brassicaceae, and we use it because they occur on multiple areas along the gradient. So that's why we're using it for the intraspecific. We do have a lot of molecular work going alongside my project. I do a lot of the environmental work. We have subsequent population genetics work going on alongside. So maybe my collaborators could answer your question in a bit more detail. I'm sorry about that.
Okay, I have one one quick question. Is glucosinolate the only chemical defense compound you know from your species, or are there more chemical compounds?
There are absolutely more chemical compounds within these species and with many other different plant species. But the glucosinolates tend to be the main thing that they're investing in. And we know we have quite a neat, we as in the general chemical community have quite a neat system of being able to look at the concentration of glucosinolates. And a lot of information is known about the biochemistry of such glucosinolates. This is why they're a good model to work on.
Yes. Okay, thank you.
Thank you.
So our next speaker is Mick Hanley. So fruits from the fire, conflicting pressures for the evolution of woody-fruited, I'm not sure how to pronounce it,
Hakeas.
Hakeas in Western Australia.
Okay, perfect. Okay, so I'll get my thank yous up first, and in particular, thanks to Byron Lamont, who's actually written the manuscript, if you'd like a copy, it's here. And to Tiu Anahi, who did all the tricky analysis that I'm about to gloss over very quickly. So, there we go. I was lucky enough a few years ago, oh God, about 10 years ago now, to get some money off the BES to jolly off to Australia to look at the interaction between floral traits in hakeas and pollination syndromes. And basically what we found in that was that if you were a bird-pollinated hakea species, there are about 150 different hakeas in Australia, then you produce these very large flowers to attract in the pollinators. The downside of that, of course, is if you're obvious and apparent to a pollinator, you're also very apparent to things like emus, kangaroos, and parrots that would happily eat the flower. So, what we found, actually quite neatly, I think, was that if the flower was very big, if the inflorescence was very big, it was laced with cyanide. And these were really high levels of cyanide, the kind of levels that we would banned in the USA as injurious to human health. The insect-pollinated species, which are the vast majority, tend to produce small white inflorescences. They're hidden away in a dense growth of foliage, which is often very, very spiny, and as a consequence, they required low levels of chemical defense. So the basic take-home message in the paper was there was an evolutionary trade-off between pollination syndrome and the need to defend your flowers against would-be florivores. So that was all very nice, but we had a nagging problem, and that nagging problem was that the genus Hakea is actually defined by the possession of persistent woody fruits. And so if you're producing these very, very apparent inflorescences, if you're bird-pollinated, then presumably the fruits are also apparent in the environment. And they could be apparent and accessible for decades, because one of the things that Hakeas do is they're serotinous, and that means they're storing seeds in the seed cone for maybe 10, 20, 30 years or more, and that resource is then potentially available to things in the environment that might want to eat those seeds. And the seeds themselves are really packed full of nutrients, so there's higher nitrogen levels in seeds than in many insect species. So potentially those bird-pollinated species are also prone to increased levels of fruit attack, i.e. pre-dispersal granivory. The main culprit is the black cockatoo. If you've ever been lucky enough to get close to one of these things, they're a very big bird, and that beak is really like a pair of secateurs. They have a fantastic capacity to destroy vegetation. And while flocks of black cockatoos are much reduced compared with 200 years ago pre-European settlement in Australia, you still see flocks of 200 or 300 or 400 birds flying around together, and when they've passed through an area, there is nothing but devastation left behind. So presumably they have over evolutionary time been a major selection pressure. They continue to be very important in that environment, but alongside the pressures of defending your seeds against fire and also the need to attract different pollinators. So we put all of that together and we came up with these various predictions. That if you are bird-pollinated and produce these accessible flowers and therefore by inference fruits, then those fruits, those woody capsules that protect the seed, should have high levels of intrinsic defense. And equally, if you're one of these strongly serotinous species that keeps seeds on the plant for maybe 20, 30 years, again, you should probably have a very, very high level of intrinsic fruit defense. And by defense, we basically mean one of four things. So that's called the cricket ball hakea. For those of you who know about cricket, that will be self-evident. If not, a cricket ball is about that big and hard. That seed, or that fruit, is very hard and very big. Equally, we could also have cryptic fruits. So many seem to be mimicking foliage and hidden away like that. Clustered fruits are probably working in the same way to mussels on a marine intertidal shore. Lots of them packed together. It means that no predator can really get purchase underneath them to extract any one of those fruits. So they're very tough, and they're also clustered together as sort of self-defense. And then finally, of course, you can be spiny. That plant on the end, Hakea auriculata, is probably the spiniest plant I've ever seen anywhere. The whole thing is probably about a metre across, and it's just a ball of pain. But we'll come back to that in a minute. So unfortunately, despite the fact we had a fair bit of existing data, largely because Byron Lamont's been working on Hakeas since about the 1960s, so we've got a really big data set on Hakea traits of all sorts. Unfortunately, I still had to go to Australia and collect some data, which was really sad, and managed to get lots of these nice sites. I won't go into details, but basically a very large number of sites across pretty much something approaching the length of Britain, all the way from the south of southwestern Australia up to the sandplains of Eneabba, which is a fantastically diverse ecosystem. And one of my main jobs was to assess levels of parrot damage in the field, just to see whether these things were being attacked in the first place. Also try to quantify clustering and the accessibility of the fruits to parrots. This is a dead parrot. We struggled with how to actually quantify accessibility. So Byron came up with the idea of getting a couple of roadkills from the museum and then modelling plasticine on the heads, and then, as he's doing there, elegantly poking this pretend bird head into the foliage, and very simply, you've got an index of how many spines will stick into a parrot's head, and in particular, these sort of important bits like the eye. So we got a very rough and ready, but actually quite neat, index of damage to a bird's head and accessibility of the fruits. Not that any parrot is daft enough to do that, as we'll see in a minute. I'm going to quickly skip over the analysis, because basically it's all published anyway. So we did the usual, have to assemble a phylogeny, because we've got 50-odd Hakea species, so we need to do all of that. We've got all the usual sort of phylogenetic independent constraints, structural equation modelling, fiddling around in art, etc., etc. The key thing is there was no obvious regional variation in levels of attack, so the parrots are causing problems right the way across the range that we worked on, and also there's no obvious phylogenetic trends in our data. So, very quickly, some quick and dirty comparisons. It's pretty self-evident, really. Insect-pollinated species tended to be much more heavily attacked than bird-pollinated species. And that's despite the fact they have much higher levels of spinescence, and also that they're inflicting more head wounds to our pretend parrot. And fruit camouflage has no effect really whatsoever in defending insect-pollinated species. Bird-pollinated species are, as you see there, 82% of them are producing very large fruits, greater than 1 gram in size. So they're much larger, they're much heavier, and also they tend to be clustered more frequently. And we managed to work out an efficiency ranking. I won't go into the details, it's a very simple index. Basically, larger fruit size was the most effective defense, better than clustering, which was better than spinescence, which was then better than camouflage. So, we can basically say that our first hypothesis has been supported. The accessible fruits of those bird-pollinated species have those better intrinsic defenses, clustering and large fruit size. The serotiny effect, well, as you might have guessed by now, it's a sort of similar story. The strongly serotinous species are producing fruits that are held for a long time, and because of that, they're producing large clustered fruits. They're providing the defense. Spinescence isn't much use, partly because you've got to let the pollinators in the first place, and also fruit camouflage. It is the size and clustering of the fruit that is really defending the seeds inside from granivore attack. So again, we can conclude that our second hypothesis has received pretty strong support from our data set. So, the take-home message is really that spines don't work, I guess, and that's evidence. So what's happened here, that's achia auriculata, that's that ball of pain, and when we were actually going around Western Australia, we found many, many plants that had literally been butchered by parrots. So what we think happens is the parrot sort of sits at the base of the plant and then sort of works upwards, and he's carefully dissecting the plant, snipping off entire branches, and you'd see, you know, a substantial amount of foliage just cast to one side, and they would work their way up through the plant, dismember the plant to get at those seed pods in the middle. And then you can basically see what they've done there. They've just snipped the entire seed cone in half with their beaks and just pulled the seeds out. So it's a major dissection activity they're undertaking there. Very, very precise. So they just work their way around the spines. So consequently, your only effective defense as a hakea is to produce these large fruits. So I guess the take-home message from all of that is not only is pollination syndrome interacting and co-evolving with floral defense and the amount of spinescence on the plant, it's also the case that subsequently fruit defense is interacting with pollination syndrome and your ability to withstand fire and produce seeds that go through to the next generation. So I shall wrap it up there, but with the caveat that my PhD student is about to start his talk and I really want to go and see him. So if you've got any questions that can wait till later, brilliant, but I'm happy to do a question now. Thank you.
Yeah, I got some colleagues who just published a couple of papers about parrots as overlooked seed dispersers. So I'm just wondering, how do you know that they're not actually also dispersing some of these seeds? It seems you're suggesting they destroy them all, but I mean, do you know that for sure?
We don't, but I can't imagine for a second on that image I just showed that they're then swallowing them gracefully. But yeah, I'm convinced they're just getting the thing and mashing it up. Those beaks are not designed for...
Yeah, I don't know. They're supposed to drop a lot when they fly away and that kind of thing. I mean, if you look at the papers by Fepiteia.
I mean, there is, yes. I mean, for sure, when they go through some of the plants, there's carnage everywhere, and they will miss, inevitably, they will miss some seeds when they're tearing stuff up. But I don't get the perception that they're dispersing away from the parent plant very far.
Yeah. I mean, I think it's a general myth that granivores actually, there are no mutualisms between granivores and plants. I mean, it's quite normal that granivores birds, not necessarily parrots, but you know, they do actually, some of those seeds are possibly not necessarily parrots, but I think it's a normal.
Oh yeah, I mean, I take that point. I absolutely agree. We don't know for sure, and they could be dispersing, but I would be surprised, just to go to all that trouble of taking those out, that they're just swallowing the thing whole. I think they just crush it. But, sure. Okay,
Thank you very much. So our next speaker is Rüdiger Reich, who is going to talk about how thermal regime drives latitudinal gradient in life history and morphology of a fish.
Yeah, should we work? All right. Yes, so I'm going to talk a little bit about, to some extent, various different environmental gradients, but the main one that we initially started looking at was latitudinal gradient. And this is data that we're just putting together now, so most of the analyses are done, but some of the things still changed this morning, so bear with me while we go through this. But basically, this goes back to the topic we've already had a couple of times today, and that is environmental gradients. And of course, we're all familiar with the fact that gradients are basically everywhere around us. They can be very small scale, they can be fairly large scale. And so we might have salinity gradients, thermal gradients, potentially even biotic gradients like predation or parasite gradients. And so there's quite a bit of evidence now from various different taxa, from plants all the way to birds and mammals, that organisms often respond with adaptive diversification along various different gradients. And one of the most well-studied gradients is the gradient of latitude, if you will, which is mainly driven by the fact that a lot of people are interested in evaluating whether organisms respond to latitude conforming to Berkman's rule, which, at least in short, posits that organisms should get larger with higher latitudes due to thermoregulation. And so while this was initially posited for endotherms, there's now quite a bit of evidence that this often seems to apply also to ectotherms. But with respect to freshwater fishes, the evidence is actually a little bit more conflicting. So there's two large reviews from at least somewhat recent years, and predominantly they found that most freshwater fishes actually seem to have patterns contrary to Berkman's rules, where organisms tend to become smaller once you get higher in latitude. And so the main caveat for these studies, however, is that they're more or less like review studies. So they looked for publicly available data from angling records and from biosurveys, and then analyzed those, which of course means that the methods of catching and measuring the fish differed from data entry to data entry, and that of course also they were from all across various different years. And then lastly, quite often, while it's always assumed that there might be some impact of temperature, not a lot of people usually actually measure temperature and other environmental variables along with latitude. And so sometimes it's not actually quite sure what might be driving certain latitudinal or other environmental gradient patterns. So here we're basically trying to address this in a life-bearing fish, the eastern mosquito fish, Gambusia holbrooki. And we're trying to look at a set of 4 different questions. The very first one is just simply, do Gambusia holbrooki have patterns that conform to Berkman's rule across their natural distribution in the US? Then specifically looking at climate and temperature, if you will. So basically, what is the effect of various different climate variables on life histories and body shape? We also looked at productivity within the distribution. So how does habitat productivity potentially impact phenotypic differentiation? And lastly, how does population density affect these patterns? So to introduce you to the model system a little bit more, this is Gambusia holbrooki, the eastern mosquito fish, a life-bearing fish where the males have this modified anal fin that they use to transfer sperm to into the female genital region. And this fish is relatively widespread across the eastern part of the US, where it's native, but due to mosquito control has pretty much been introduced now almost worldwide and is one of the most invasive fish as well. So in September of 2011, we sampled within a 10-day period basically the whole natural distribution along the Atlantic coast of the US, starting with a population in southern New Jersey and then all the way down to a population on the westernmost Florida Keys. And at each site, we try to estimate some environmental parameters, including population density and chlorophyll A, which we used as a proxy for habitat productivity. We then extracted additional climate data from the World CLIM database, and then included all these together with latitude and longitude and a couple of other environmental variables, first into a principal component analysis to kind of reduce our environmental variables, and ended up with 4 environmental PCs that we then used for our analyses. With respect to the actual data that we collected, we employed fairly standard life history dissections to get a set of somewhere between 6 for males and 7 to 8 different life history traits for females. We then also conducted body shape analysis based on geometric morphometrics, using x-rays of the same individuals that we used for the life history dissections. And then all of this went into a model selection, model averaging approach to kind of try and tease apart which environmental factors might be driving certain phenotypic patterns. So let me walk you through some of the results, and this is where some of the new data from this morning come in. So first, let's have a look at some of the patterns along our PC1. I showed you all the environmental factors that are kind of like loading either positively or negatively onto this PC1. But as a quick summary, basically PC1, if you increase along PC1, you're increasing latitude, longitude, and basically get into colder, more variable climates at the same time. So along this axis then, if we first look at the first measure of size, so here standard length, we basically see females here on top, males here on the bottom. We see that in both sexes we actually get this pattern that is predicted by Berkman's rule, where fish tend to get larger with increasing latitude. But at the same time, of course, we also know that this seems to be mainly due to colder, more variable climates. Then looking at a second measure for fish size, in this case wet weight, we basically find the same pattern replicated. So again, two different measures of size, but both indicate that Gambusia holbrooki tend to become larger towards the north. And then we looked at a couple of other life history traits as well. And so the next one, before I show you the actual pattern, let me just walk you through real quick. So basically we compressed what we can call investment into reproduction for males and females into one graph. So in males, that would be the gonadosomatic index. So in other words, the relative weight of testes relative to the whole body weight. And in females, that's the weight of the whole clutch relative to the whole body weight, okay? So you then basically get a fraction that you can use to estimate how much they invest into reproduction. And this is shown here. I didn't have time to change the axis label, so the GSI gonadosomatic index in this case is supposed to mean both the reproductive allocation, as we call it normally in females, and the actual gonadosomatic index here for males. And we can see also that there's a nice positive trend where investment into reproduction increases with latitude and colder, more variable climates. Now looking at some of the other traits, in this case along PC3, so the nice one with PC3 and 4 was that they only had one environmental variable that loaded really high onto them, so it makes it a lot easier to interpret what might be driving this. So in this case, it's basically productivity measured by us as chlorophyll A. And if we get into more productive habitats, we also get this increase in size measured as weight in these fish. And at the same time, not surprisingly, the fish also get into better body condition measured as body fat. So the more resources are available, or the more highly productive the environment is, the more fat the fish are able to put on. And then when we look at one pattern for PC4, which only corresponds to density, we find that now, again, this is investment into reproduction. We now find a decreasing pattern, even though it's much stronger in females, where with increasing population density, investment into reproduction is actually decreasing in Gambusia holbrooki. So what does this all mean? Hopefully, I can walk you through this. So the first question was about Berkman's rule, and we find actually clear support that these fish conform to Berkman's rule. They become larger in higher latitudes. But we were basically able to show that at least for these fish and the variables we measured, this seems to be more a result of colder, more variable climates in the north. Now, why did some of the previous studies that I outlined before, in particular for freshwater fish, potentially not find this, or actually even found the opposite pattern? So in one of the reviews, a close relative, the western mosquito fish, was used as well, and they found a pattern that is opposite to Berkman's rule in those fish. So again, I think it's largely due to the fact that they actually didn't sample specifically from one particular point in time, but actually just used various different available data to compile their data set. But in particular, actually our data set, which was kind of almost a coincidence, but we sampled in late September 2011, which basically means that for the northernmost populations, it's very close to the onset of the first cold spells. And so if there is a pattern where, as we assume, where body size in this case is actually largely responding to overwintering survival, which in fish has been shown that larger individuals have a higher survival probability across cold, prolonged periods in the winter, then we would actually expect to find this pattern late in the season before the onset of winter, but potentially even in Gambusia holbrooki, if we had sampled a couple of months before, potentially we might not have actually found the same pattern. Then with respect to climate in general and life histories, some of this I've shown, some other things I haven't. So reproductive investment is actually increasing and offspring size is decreasing, and we have a little bit of a tendency for fecundity to also increase if you go into higher latitudes and into colder, more variable climates. And we assume again that this is actually due to mortality. So probably what happens is that in these more northern populations, climate actually changes sometimes quite erratically even through the winter, through the summer and spring. You might get occasional cold spells. Potentially there's also spikes in nutrient and resource availability. And so probably because of that, the fish are selected to invest as much as possible into individual reproductive bouts because there's relative high uncertainty that they might survive to a subsequent reproductive bout. And at least this pattern would be congruent with this. We also found some interesting patterns with respect to how body shape reacts to climate. I'm not going into detail here because the very interesting result that we found was that almost all of the body shape variation with respect to climate seemed to have been due to covariation with life history. So once we included some important life history traits as covariates into the model, the body shape effects completely disappeared. And we can talk about that later during the Q&A or even after the talk if you want to. Now, what about productivity? So here we found that fat content and body size increases together with deeper body shapes in high resource environments, and of course that kind of intuitively makes sense. If there's more resources available, the fish are actually able to use some of the excess resources to invest into body condition like body fat, which would then secondarily also make the bodies deeper. And then lastly, with respect to density, mainly investment into reproduction and fecundity actually decreased in higher population densities, which seem to again have been linked to competition, in this case of course due to density, but actually affecting how much resources are available for the individual fish, since there was some covariation between productivity and density as well. So what is the take-home message then? Well, the first one is that we did find support for Berkman's rule, and that actually we did find some good evidence that this is linked to thermal regime in this species. But then even more importantly, we found some more evidence for, of course, kind of the obvious effect that multivariate phenotypes like whole suits of life histories and body shape are usually the result of multifarious selective regimes. So in other words, just picking one or two traits and one or potentially two environmental variables among the plethora might yield you some interesting results, but you might actually be overlooking some other factors that are going on in the background, and that sometimes certain selective agents might actually synergistically either work together or antagonistically work into different directions or potentially even cancel each other out. So it does become quite important to actually look at all of this in a more multivariate setting. And the very last one, also a really important one, just because you do find some correlation between certain traits and environmental variables doesn't always mean that there is direct selection on these traits. As we showed with our body shape analyses, they can be due to covariance to other traits that are actually under selection. So with this, I have to thank all my co-authors and the different institutions and departments and ministries that actually issued the collecting permits, and hopefully I can answer some questions. Thank you.
So we have time for one quick question. Yes, please.
I don't know if you know a paper about predation by waterbirds in the Tamar.
Mm-hmm.
Suggested that predation by herons, for example, can affect size distribution of Gambusia populations.
Right.
Sex ratios or male bias.
Right.
Did you think about looking at predation gradients as well, or how that might?
So in this particular case, we were simply not able really to do that. So the main thing was because a lot of these sites we went to for the first time, even though we tried to look for sites where people had collected them before. and we did a little bit of like what other fish were available, but it seemed to have been way too spurious because some habitats were more easily accessible to catch other fish as well. Some others were not that easily accessible, so maybe we didn't catch the actual predatory fish that might have been there, or potentially other birds, of course, if you just do a quick scan sample. So I'm not necessarily familiar with that particular study, but I know, of course, there's like other work from the US too. Joel Traxler has shown that herons are usually size selective in their predation rates. So we most certainly know that we didn't measure everything, and it is actually quite likely that there might be some patterns in here that to us look as noise, and they might be the result of high or low predation at various sites. But of course, various other factors as well. So salinity we didn't really specifically look at. So yeah, I'm pretty sure there's a lot with predation going on in here too, but we weren't able to directly test for that. Mm-hmm. Thank you.
So our next speaker is Harry Marshall, who is going to talk about lifetime fitness consequences of early life ecological hardship in a wild mammal population.
Great. Thanks very much for coming. As I said, my name's Harry, and I work as part of a group studying wild banded mongooses in Uganda. I'm going to talk a bit about some work I've been doing today, investigating the importance of early life ecological conditions on their reproduction and survival. So, just to kind of illustrate the importance of early life environment, this is my nephew, aged -2 months. And then this is him aged 2 years old, and one of the things we might ask is, why is he making those stupid faces? And is there something terribly wrong with him? And the answer is no, it's just that his mother's also making very stupid faces. And so, in a slightly flippant way, but that's kind of illustrates that the environment you experience in early life can influence you in quite a profound way. And if we think about perhaps a slightly more serious way, there's actually now quite a plethora of studies to illustrate the importance of particularly the ecological conditions you experience in early life on a whole suite of aspects. So on growth, so we know that baboons grow faster when they have a more favorable early life. Behavior, survival, reproductive success, and particularly, for example, the trade-offs between survival and reproduction. So there's a lot of theory and a lot of work coming out now to suggest that the quality of the environment you experience during development should influence the amount you invest in survival and reproduction in later life, in some kind of life history trade-off. However, many of these studies, either implicitly or explicitly, really they focus on the average environment being experienced in early life, and that's obviously very important. But what I've been trying to ask a little bit is, what about the variability of this environment? And we know that ecological variability should be important, and there's a lot of talks in this session been about this. And in particular, for example, that the variability of environment should determine which of various different life history strategies is more profitable. And bet hedging theory would tell us that actually in a more variable environment, often the low variability strategy is more profitable, even if its mean return is lower. And there's now some work which is starting to suggest that the variability of your early life environment is also important. And in particular, I'm going to highlight here some theoretical work by Rebecca Hoyle and Tom Ezard, and actually also Bram Kuiper is not on this paper, but there's a whole suite of papers that they've published in the last 4 or 5 years. And they have been looking at how particularly maternal effects, so the correlation between mother's phenotype and offspring phenotype, what type of that maternal effect is favoured under different levels of environmental change. So in this example, they simulated a environmental shift where the optimal mean phenotype shifted, and found actually a moderately positive correlation between mother and offspring, allowed the population to reach this optimum optimum phenotype more quickly. And actually, the scale on here is thousands of generations. So even though it looks quite small, that's actually quite an important difference from the black line, which is when there's no maternal effect. In contrast, when the environment is stable but varying around some mean, then actually this favours a negative correlation between mother and offspring. So this is some sort of stabilising effect. However, the nature of the variability, so this is represented by these different lines, determines exactly where the maximum fitness return is. So these example just really serves to say that the variability of your early life environment is important. That should have an important effect. So as I said, I've been looking at this in banded mongooses. This is a long-term project based down here in southwest Uganda, and on this peninsula here. So almost an island population. There's a very just tiny little isthmus connecting it to the rest of the land. And this is a population that has been studied for 21 years now. We have full life history information, over 3,000 individuals, and at any one time there's about 200 individuals in 8 to 10 social groups. And these are them. So these are banded mongooses, and they, just a bit of background, they are highly social. You can see they're sleeping in big piles, and live in groups of 10 to 30 individuals. And I guess the important natural history information for this talk is that, unlike many other cooperative breeders, they all invest in reproduction to some extent, and they all invest in cooperative care to some extent. So there's quite a large variation in both in reproduction across the group and survival. And in particular, I've been looking at the importance of the rainfall they experience in their first year. So this is their first year is their main developmental period. This is when they do almost all their growth. After a year, they tend to sexually mature and start to reproduce. And looking at both this is the sort of 12-month running mean of the mean and standard deviation and monthly rainfall on our site. And nicely, they're relatively uncorrelated. They're only correlated to about 0.3. So we can put them both into a model and decompose the effect they have on various life history parameters. And asking, how does the mean and the variability of these conditions influence individuals' survival and reproduction in later life? So we look at the rainfall in their first year and look at how this influences their survival and reproduction in adulthood. And very briefly, I'm not going to go into this in a huge amount of detail, but firstly, is we find that there's almost no effect in females. So female survival and reproduction is completely unaffected by their early life environment, but there is strong effects in males, and this is what I'm going to focus on now. If you'd like to talk to you more about the sex differences, I can talk about it afterwards, but there's not really time to talk about both these results in this talk. So let me walk you through these plots. So this is the effect of the mean rainfall in early life on various aspects of survival and reproduction. So you're going to see two sets of these plots. And this top plot here is showing the effect on lifespans. This is in years. The second plot here is something I've called relative fecundity, and I'd be really happy for any suggestions of a better name, because I don't really like it. But it basically shows the proportion of all individuals that a male could have sired, so all pups that were born into the group during their lifetime, the proportion that they actually did sire. And the final plot down here is much simpler. It's just simply the number of individuals, so lifetime reproductive success. And what we find is that mean conditions seem to have quite a classic life history trade-off. So the difference in the pink and the black here is the pink individuals are those who actually successfully bred during their lifespan. So these are the only ones who go forward to the second plot. And in those individuals that did breed, the individuals who had a good early life, so had a high amount of rainfall, which we know is correlated with food abundance. They had a shorter life, but they actually sired a higher proportion of all individuals they could have, compared to these individuals who had longer lives, but a very low proportion of all pups. But obviously they had more pups to sire because they had a longer life to live across. And ultimately, this needs to no effect on lifetime reproductive success. So there seems to be a trade-off here in investment between reproduction and survival. As I said there. However, if we then consider, this is just looking at the average conditions and individual experience during their early life. If we now actually look at the other side of this coin, look at how the variability of conditions in early life affects survival and reproduction, what we see is a much simpler but perhaps slightly more puzzling effect, where actually if you experience a more variable early life, so up this end of the scale, you just live longer. And there's no effect on this relative measure. And the ultimate effect is that you sire more pups. So you seem to have a better return. And this is a bit puzzling because we know that actually more variable conditions is associated with lower food abundance. And similarly also rainfall influences body masses quite strongly. So you'd actually probably predict, classically you might predict in the opposite direction. So why might this be? Why do these more variable conditions seem to produce better mongooses, for once a better expression? So we've tried a few different things to try and understand this effect. So perhaps is it selective disappearance in the first year? Are these harsh conditions just weeding out all the rubbish individuals and we're left with the good ones? But no, that's not the case. There's no effect on survival in the first year. Is there some sort of rank advantage? We know that older, high-ranked females are more likely to breed under poor conditions, and low-ranked individuals only get to breed when it's good. But no, there's no effect this as well, or any effect of the amount of social care these individuals receive. As I said before, we tested in these models for some sort of trade-off, but didn't find that. So this kind of leaves us with a hypothesis. We don't really have any direct evidence to support this, but some idea perhaps that these individuals from more variable early life are better equipped to deal with a wider range of environmental challenges. And there's kind of some literature that might tangentially support this for something like human health. So we know that if children who experience a very clean early life and don't experience so many pathogens, potentially have more allergic response in later life. So you can perhaps see a sort of similar argument being made in the human literature. But this is really an open puzzle that we're still trying to puzzle out. But we seem to have crossed off some of the main possible explanations. So in conclusion, this is sort of just a figure to try to sum up what we found. These is sort of trying to represent the life history trade-offs between that we find. So each of these thin lines here represents equal fitness return, and the dotted line represents sort of the reference trade-off. So when a change in mean conditions basically results in a shift in this line, and a change in the optimum investment in trade-off between reproduction and survival. So better conditions, you invest more in reproduction, less in survival, and vice versa. However, a change in the variability of early life conditions seems to actually just relax this constraint. And so more variable conditions seems to allow you to just have greater fitness returns. Essentially, as far as we can find so far, no cost. That doesn't really seem to make any sense, but so far we haven't found that. And I'd just like to finish by saying thank you very much to Francis, Kenneth, Robert, Solon, and Solomon, who are our great field assistants in Uganda. These guys for funding, and you for listening. Thank you. Yes, so when we did one of that in the paper which we were just preparing at the moment, that is in our discussion is one of the possible arguments. There's obviously quite a lot of these stuff, many from humans and lab systems in mice, that show that one of the really consistent ways of increasing lifespan is to reduce to a, not completely, but a mild level of calorific restriction, which could definitely explain some of this, because these animals live in quite an equatorial system. So even when conditions are bad, they're bad enough to affect survival and weight, but we don't see massive die-offs. I suspect with some of those studies as well, they may not have, because they've been in lab systems, they haven't really been able to look at trade-offs. Those systems aren't subject to natural levels of selection, predation, pathogens, and so actually, if you repeated those same studies in a natural system, you might find that, although they're living longer, they're actually having a lower per unit time reproductive success. So perhaps like this, the mean effect of mean conditions on the left.
One more question? Yes?
It seems that you have an increase in individual variability in reproduction with increasing mean rainfall. On one graph, you have very little effect on the mean, but you have quite marked increase in variance. Yes, in this graph.
That one?
Yes, the previous one.
That one.
This one, yes.
Mm-hmm.
Relative fecundity, the variance is very high at the highest.
Up here. Yeah.
And so, do you have any interpretation of the mechanism that would cause that?
My parsimonious suspicion would it be it's simply an artifact of the fact that this is a proportion. So as the mean will increase, then presumably you will get a greater amount of variability simply due to the way that this works. So it's a probability, it's a proportion. So a bit like I think you were in Jenny McDonald's talk this morning. She's talking about the relationship between mean and variance. My gut feeling was simply it's because the mean is getting higher, and we expect the variance to get higher simply due to the mathematics of a proportion. Maybe there's something more interesting going on, but I think it's quite difficult to discount that slightly less interesting but explanation.
Thank you. So our last speaker of the session is Christopher Hassel, who's going to talk about ecology and evolution of mimicry in a changing world.
All right, well, thank you very much for sticking around, and I apologize for keeping you from your poster-related alcohol. I'm going to talk to you about some results in a fairly superficial way across a project that I've been running for the last 2 years while I've been a Marie Curie Research Fellow, but it's one of those projects that's been gestating for a number of years before that, so it's nice to finally be showing some results to some people. So I'm going to be looking at mimicry and how that ties into climate change. Just because we haven't had any mimicry talks as far as I know yet, I'll give you a quick primer on Batesian mimicry, which is the sort of mimicry that I'm going to be talking about. There are a series of premises. Some people might disagree with the way that I phrased this, but this is the way that I'm going to phrase it. Basically, prey vary in their level of defense. Some prey are defended and others are not. If prey have a defense, then there is a tendency for them to evolve some kind of signal by which predators can learn to avoid that defense. We call that aposematism. We then have a series of undefended prey in the community that have a tendency to evolve similarity to that signal. We call those the mimics, so we have the models and the mimics. The predators gain by obviously avoiding the harmful models while exploiting the harmless mimics. And the discrimination between those different types of prey occurs through a series of iterative interactions with the prey community, whereby the predator learns to discriminate one from the next. We can make a series of predictions from this rather simplistic explanation of Batesian mimicry. First of all, that models, that is the defended prey, would benefit from rapid learning by the predators. The second is that the mimics also benefit from rapid learning by the predators. And the third is that predators also gain from rapid learning by the predators, assuming various things about the nature of the prey community. So for all three actors in this system, we might expect that the optimal sequence of encounters would be a series of defended models first for the predators to be educated, followed by a series of mimics off into the distance once that learning has occurred. So if that's the general evolutionary context within which we're working, how does that relate to what actually happens in the field? And how might the spatiotemporal variation that's promoted by climate change disrupt the kind of optimal sequence of encounters that predators and models and mimics each need in order to gain the greatest benefit? So I'm going to talk first about a study about the order of presentation and occurrence in the field. We have reasonable expectation that climate change is going to be influencing one particular model system, and it's the one on my title slide. It's the syrphids, the diptera, hoverflies. We know from a series of laboratory studies that they're very sensitive to temperature in terms of their development rate. So these are studies collected from the literature published earlier this year in Ecography. So we know that they respond very closely to temperature. This gives us a mechanism by which we might expect a phenological shift in the mimics in this case. We also know that when you look at the sum total of biological records collected in the UK, and we're very good at collecting biological records, that we see a very strong relationship in the field with climate change. So highlighted in yellow is the diptera, the majority of which are syrphids because they're so well recorded. And the graph simply shows the rate of phenological shift, or the strength of phenological shift. So it's the correlation coefficients between temperature and flight dates. So a strong negative correlation means that they are responding strongly by advancing their phenology, or occurring earlier in the year. And that's compared against a series of other UK taxa. Also featured on that plot, of course, are the Hymenoptera, which are the models in this system. The majority of those are bees and wasps, but there's a series of ants, which we'll throw out for now. So we have, in an aggregate at a community level, our models and our mimics, which are both responding very strongly to climate change. Note that the Hymenoptera are not responding quite so much, but we don't have to settle with this community level data. Because mimicry has been so well studied in these two groups, we can look at pairwise interactions between models and mimics to see if they're reacting to climate change in the same way. Here's one example out of hundreds that I could pick out. In red, we've got Bombus terrestris, the very common bumblebee, buff-tailed bumblebee. And in black, on the right there, that's Cheilosia illustrata, the mimetic hoverfly. What you see here is what the textbooks would tell you about how Batesian mimicry works. You have very early emergence from the effectively winter-active bumblebees, followed later on in the season by the hoverflies. So they're occurring later in the year, so they're further down on the graph. The months are to the right, just to aid in interpretation. But they're both tracking climate change in a very similar way, so there's no difference between those two slopes. What we can do, because we know a lot about the model-mimic pairing in these communities, is we can then take these correlation coefficients, we can plot one against the other for each of all the possible combinations of models mimics that we can look at, and we get something that looks like this. So just to guide you through this, from left to right we have the leading edge of the flight period, so this is the start of the flight period early in the year. The center is the median flight date, the middle of the flight period, and the trailing edge on the right. At the top we have this Pearson correlation between the flight date and temperature, and on the bottom we have the correlation between the actual numbers of days shifted and temperature. So it's a rate of shift in days per degree change. I want you to take two things away from this, one of which I'll talk about and one of which I won't. The first is that there's a big difference between the different phases of the flight period, that we get a very strong relationship in both groups at the leading edge of the flight period, which decays towards the end. But more important for what I'm going to talk about is the fact that we have a lot of noise. And as we know, evolution works on noise. So there's an awful lot of variability in these systems, and some model mimic pairs are tracking very nicely. They lie on the one-to-one lines on each of these plots, but there's a lot of species that don't, a lot of pairs that don't. So it's those that are of interest. So people have been looking at biological records for years. I've stood up here last year, in fact, and talked about, at BES in Edinburgh last year, talking about hoverfly phenology. But what does that mean in terms of the evolutionary links between these taxa? So I took two complementary approaches to try to figure out the answer to that question. The first used virtual reality, because virtual reality is cool. I've got a really great collaborator, Jack Billington at Leeds, who's we've been working quite closely for the past year, and we've been building these computer games, producing immersive environments for human participants to act as predators to look at selective pressures. So that's my very patient PhD student, Gio, who's demonstrating in the corner there. And what we did was we presented the participants with different types of insects based on a series of different model mimic pairs. These are your traditional, if you work in Batesian mimicry in this system, you know you've got your basic honeybee mimics, your wasp mimics, and your bumblebee mimics. So we presented one example of each kind within the experiment. We also presented, and this is important, 3 different phenological scenarios. So we presented the model first, so our stinging insects first, and then, which is the theoretical optimum. We then presented mimics first, which is the theoretical suboptimal strategy. And then we presented them in a random order, so they were interspersed amongst one another. So what I'm going to show you is the rate of predation relative to this optimal strategy, the model first. And what we would expect is that the mimic first would result in greater predation for all individuals involved. And that's pretty much what we see, actually. So this turned out as we'd expect. The one line there is, so the horizontal reference line is the hazard rate or predation rate from the model first experimental runs. On the left, we have the mimic first, where on the left plot, we see the mimics have a far higher predation rate. And on the right-hand plot, you can see that the models also suffer as a result. So you're seeing costs to both models and mimics when the mimics are presented first, exactly as we would expect. So the predators, our human participants, are having to unlearn a preference for a profitable prey when the harmful prey comes along. The random, in this case, the mimics don't really care either way, whether they're random or model first, but the models actually also lose out when they're interspersed in amongst the mimics. So that's presumably because the predators, our participants, are struggling to form those associations between the different morphotypes. The interesting thing is, because the game was structured in such a way that people were trying to score points, they gained points when they ate an insect that was harmless and lost points when they ate a stinging insect, we can actually look at the sum total of the points that the predators accumulated at the end of the game, and that becomes a proxy effectively for predator fitness. And I don't know whether you can read the plot at the bottom, but there were negative effects of both alternative scenarios compared to the model first. So the predators did worse when the models weren't first, and that was significant in the case of the random scenario. So we've got some pretty good evidence from a completely ecologically irrelevant but very tightly controlled experiment for a potential selective cost to shifting around of the order of models and mimics. So the next question then is, what does this mean in the field? So we went out and we did a series of pastry prey experiments. These are wonderful fun and extremely time-consuming. And if you doubt my handiwork in creating some of these animals, Steve Sait's Venturia canescens tried to oviposit in these multiple times. So I reckon that they're doing pretty well at trying to be little insects. And these were deployed out in the field at a series of locations in groups of 24, so 12 of each kind at a series of sites. Often these sites they're only conducted at a small number of sites. So fortunately, I had some eager collaborators, Chan Ku Kang and Amy Gresham, who conducted the same experiment at Ottawa and in Nottingham, respectively. So we had a series of sites, an awful number of baits to make and analyze. We presented them in the same way as we did for the eye tracking experiment. And I'm going to show you now the comparison with the previous study. So I'll show you the same plots as for the experiment with the eye tracking. And it doesn't work at all. So I'm being very open and honest with this. There are some issues with trying to get to grips with this at a field scale because it's a really complicated system to try to replicate in the field. We had a lot of predation that was all over the place in terms of the frequency of predation versus not. There's a lot of data cleaning that needs to be done. So I don't have too much faith in these results anyway, but it suggests this is going to be a nuanced pattern if it's there at all. So in conclusion, there's good reason that we should expect some kind of decoupling or at least shifting in the relative timing of models and mimics within field systems. Our biological records analyses suggest that that's going to happen, at least in some cases. The sort of tightly controlled experiments that we're doing in VR suggest that you can quantify significant costs to reordering away from some kind of optimal sequence. And that might be negative for all three participants in the evolutionary system. However, trying to get a grip on this in an ecologically valid field setting is really complicated, and I don't know that it's going to be feasible to do it at the scale that you'd need in order to control for all the confounding factors. But I might give it another go if I can find a lot of very enthusiastic undergraduate students. So that's all I have to say. There's a lot of people involved for the biological records, Jack Billington especially, and Tom Sherritt for the initial conversations that spawned the project, recording schemes. Many thanks to the EU, long live the EU, and thanks to everybody who's helped out along the way. Thank you very much.
So one quick question.
wind time. Did you have a question? Yep.
So I guess the predators, they in many cases are birds.
Yes.
They might actually live longer than a single season, so then the whole premise of the whole hypothesis falls apart, doesn't it?
Well, that's not always true. So there are often situations where the fledglings are the ones who are exerting the greatest costs and benefits on the different mimicry systems. There's a lot of studies done in the 1970s in the US around Chicago. Wildbauer, I forget his first name, who looked at fledgling phenology in relation to the phenology of models and mimics. Those are basically what we're aiming for in terms of the framework for the study. That there are going to be naive birds. There are always naive birds of varying extents.
Okay, thank you very much. So we